lead optimization. These things are more fluid than that, but just for this talk, we're just sort of breaking it down into a more simple stages. So from hit to lead, basically you want to figure out what have I got in my hits and can I move it forward. And if you've got a hit from a library, and this is a very poor example, but you see that you've got all these different circles, different colors. You don't know, if this is your hit, which of these circles, which part of this molecule is important. You don't know. Could it be just one piece is important? Could it be two pieces? You have no idea. So in the hit to lead stage, basically what a chemist is going to do, they're going to say, okay, I've got my hit, or I've got a natural product, or I've made something from the patent literature. What is the key essential scaffold here? So basically you have to break things down, figure out what is important, and then once you know what's important, you can build back out and explore different chemical space. When you start with fragment evolution, or start with a fragment, it's a little bit more straightforward to see, because when you start with a fragment, that is your scaffold. That is the part that is binding. So you don't have to cut off a lot of the lettuce around it at the beginning. But then what you have to do is, most likely this has an IC50 of something in the millimolar or something insane. You need to actually get activity by building outwards from your fragment. So there might be more than one fragment you're working with. You want to have more than one shot on goal. You might have a couple scaffolds you're working on, but you want to build out to expand from the core and move from binding to activity. And just a sort of a graphic to show what this might look like if you have a picture of a protein and then your fragment is binding in this spot. The goal is to build out from that spot. And as Laura said, you're going to increase molecular weight as you do this. You're going to increase lipophilicity to some extent when you do this. You're going to increase all those properties that you want to keep down. So you want to always be cognizant of that and not go overboard. But you want to, this is why you want to start small, because you're always going to have to add mass. So overall, for a hit to become a lead, it has to show structure-activity relationships. And the activity should be sensitive to structure, because if everything you do changes nothing, then you're going to have a problem. One thing I really want to point out is that losing activity is not a negative result. If your chemist makes a few molecules from a hit, and you look at one and it has absolutely no activity, that's not a bad thing. That means that you just found something very, very important. Because if something does not work, then you have to put it back because it was important, right? So just as a reminder, when you're looking at these negative data, right, it's hard to say good or bad, is not a bad thing at all. It's actually narrowing the chemical space that you have to work in, because now you know what's important. And that's really a key factor in early stage discovery. The compound should have handles for reactivity, and that means chemically. As far as synthesis, you have to be able to modify them. Most of the time, scaffolds are retained during optimization, so you want to start with something that's good to work with. You can make very complicated scaffolds with lots of stereocenters, but stereocenters do equal cost. And you might have to add in complexity later, but once again, it's one of those, well, do you want to start with complexity and have to take it out, or do you want to have to add it in if you need it? And I'm sure there's opposing camps on that one. Also, you need cellular assays, or in vivo if you can and you have the models. Can your hit get to where it needs to be? Is it brain penetrant? If you're working on CNS, you need to find that out. You need to find that out as early as you can. And you need to have these lead-like molecular properties, which will really just expedite and simplify optimization as you move forward. So in lead optimization, that's the chemist, and these are all the things you need to think about. And it's a little bit overwhelming, because now you have to think about bioavailability, you have to think about herd channel binding, drug-drug interactions, CYPs, everything. However, from what you've been hearing for the past day and a half, if you build a lot of this in at the front end, this is going to be a lot less daunting, because you have the molecular properties that will help you navigate a little bit better past this group. So the key here is in vivo efficacy. And really, it's this medicinal chemistry refinement. It's this recursive process that Marty was talking about. It's sort of you synthesize compounds, you screen for activity, or you're screening against activity for something you want to avoid, or you're screening for molecular properties, or you're doing all of this at the same time, hopefully, and generating a lot of information. You get data. You see trends of structure activity. You want to refine that. You have your interdisciplinary team sit down together, talk about these data, think about what is next, and then think about, okay, well, what can I make next to address some of these issues and sort of lead us in a better direction? And then it goes on. And it's a recursive process. So in order to do this, you do have to make a lot of compounds. Maybe you only have to make under 100, but that's still a decent amount. So just some things to think about is that generally compounds are made in bunches and not as single entities. So you want some sort of synthetic platform that your chemists can work with, that they can make molecules sort of quickly. The more molecules that are made at once, the more you can understand overall trends in efficacy, physiochemical properties, et cetera. And that it will also help because if one compound fails to show the in vivo pharmacology that you want, there are other compounds to fall back on. It can answer that question. Well, is it my chemistry or is it actually the target that I'm working on? And it might not give you the answer, but having more compounds will give you a lot more confidence in your decision making. Compounds also that might be very similar in activity have very different ADME properties or selectivity, and you don't want to cut yourself off by focusing on one at a time. You want to make series of them to spot trends, and that will guide future research not just for this project, but may even extend into other projects. So parallel synthesis of groups of compounds made by pretty facile reactions from a common intermediate is kind of the way to do this so that you can generate a lot of compounds, then generate a lot of data, generate a lot of information, make plans to make your next cycle of compounds. So just a quick, very basic case study of, well, let's say this is my scaffold. I've got 3 handles of activity, 3 areas on this one ring that I can interact with. So the first thing you could do, for example, is take this compound, and you've got a carboxylic acid here, and you can do amide coupling like you see in peptides and put them together. And you can commercially put a lot of things in this position. So you can explore a lot of different space, and these chlorides will not react in those reaction conditions. Then you can take the more reactive chloride and put various groups. You can make carbon-carbon bonds, and obviously I'm not going into the details of the chemistry, but just sort of the idea of then you can explore various space in this one position. Then you still have that one chloride left, and that chloride can, for example, you can put different heteroatoms there, different nitrogens, different oxygens. So you make some, then you test it for all these properties, you get the data back, you see, well, this is a dead end, this doesn't help activity, this seems to help, and then it's this recursive process that keeps going on until you refine down to a few compounds that you want to move forward. And then once again, there's so many reagents out there that you can put in all these positions that you need to prioritize because chemists are like, oh, look at that, I can order 55,000 of these things and make tons of compounds. That's not necessarily the goal. It's the smart chemistry idea of saying, okay, you can make everything, but what's important? So just for my going over for the last couple minutes, I'm going to just shift gears completely to touch on something that Marty talked about a little bit, stepping away from the early drug discovery process and just thinking about, okay, let's say everything works great, you've got potentially a clinical candidate, and now you need enough of it to go into rat toxicity models and potentially moving forward into humans. How do you shift gears? Because I'm saying make lots of, little amounts of lots of things, make lots of compounds. When you want to just make one, how do you go over that? So that's really the synthesis of an active pharmaceutical ingredient. And you have to make sure these things are scalable, up to kilogram scale. And you want syntheses that avoid obvious toxins such as palladium, which are very useful and very common in early stage drug discovery chemistry. Removal of metals can be very expensive. So once again, if you can avoid putting them in, that's better. You want syntheses that can be purified easily, and then salt forms are often used as APIs because they're more stable and soluble. So just a quick case study to finish up, and this is for an API synthesis. So this is the discovery synthesis. So as I said earlier, you've got something where, okay, I've got 2 chlorines. Now I can put lots of things in one position, lots of things in another position. I can look and see what my structure-activity relationships are, and I can make lots of compounds. But in this last step, you actually are using palladium to link these 2 pieces together. So let's say you decide, okay, I'm going to go with a methyl group here. This is the compound that we want as our clinical candidate. You don't want to keep this synthesis, because then you have palladium in your last step. So you want to change things around. So basically, what we have here is the synthesis of the methyl compound as an active pharmaceutical ingredient. And as you can see here, in the details, there are no metals in the last step. Actually, the last step is actually done in water, which you can think of as being green chemistry, which was not actually what we weren't trying so hard to be green, but we were trying to come up with the last step that could be really easily purified and be done in water. I mean, it doesn't get much better than that. So you eliminate the metals, you eliminate all that, you have something that you can scale up to kilograms and move forward with. So it's a completely different thing from drug discovery. It's something that you'll most likely be outsourcing, but it's good to know the factors involved and the things you should be thinking about if you're able to make that step forward. So just a summary and conclusion. Basically, the path to drug discovery begins with the selection of the compounds or the library that you're actually screening. The library should be chosen for the same reasons that compounds are chosen later in development, all the things you've been hearing about properties. There are a lot of complementary ways to get hits. Patents, screening, different types of screening. Optimization, you're looking at an increase of potency and selectivity, but you really want in vivo efficacy. That's what's most important. Once you have your potency, you can optimize other factors. And looking at drug-like molecular property filters really facilitates this process. And you know, for your chemists, they're going to use this pattern recognition and parallel chemistry to really move things along. So overall, it's a very complicated process, as you know, I don't need to convince all of you. But the key is that having drug discovery chemistry, you're working to find these compounds that really are going to hit in a sweet spot and give you the best chance possible to get a drug. Chemistry efforts are really led by biological results. So chemistry doesn't happen in a vacuum. Biology doesn't happen in a vacuum, which is why this constant communication and these formations of interdisciplinary teams are really the key. And as Marty said, academics, you have an advantage over some bigger pharma where people are separated. You sit across the lab bench from your person who's doing the, your in vivo studies. That's how I had it. It worked out great. So communicate. Take your resources and share them. And then you have the best chance to overcome the many obstacles and succeed in the discovery of an efficacious drug. So thank you very much.
So one of the things that was mentioned here, and I think to some extent in previous talk, is the selection of the library and selection of the compound for screening, and the desirability of certain physical chemical properties that would make it more drug-like. So I'm curious in your thoughts as to how one balances correctly the desirable to narrow things early on in the screening that are as much drug-like as possible, yet doesn't make it so stringent and doesn't rule out of things that might not be as desirable, but it could be modified later on to have the desirable properties. And maybe a similar question to that is how much can one really, through chemistry efforts, modify such properties later on?
Well, I guess this is also more of a general question to everybody, sure. if you guys want to weigh in. I mean, it's a good question. I mean, it is a balance, but the idea is that if you start out, it's always easier to add in complexity. It's easier to add in molecular weight. It's easier to add in grease. When you start with these molecules that are greasy and larger, and they might be diverse and they might look pretty, but they might just be non-selectively binding to everything. So, you can always it's much easier to put things in, like Laura said, than to take things out. So it's really just, but it's also knowing your target. So if you're working in a space that, doesn't really comply with these limits, you might have to look at more at patent literature. You might want to look at natural products. It's also knowing what your project is and what the correct route is, because high throughput screening isn't necessarily what you'll do for everything. Or I think it very much is project dependent. Yeah. Right, you can't have compounds that are only going to bind if you're looking for activity as a readout. So, obviously you'll need things that start off a little bit more complex and bigger, but you don't want to start out with things that have a molecular weight of, close to 500 when you're just starting out. So you're not going to want something that has a molecular weight of 150 because you might not get your activity, but you want to think about all these things in the context of what you're looking for.
morning. I want to thank the organizers for the opportunity to talk to the CNS community about natural products. It's not something that we often do. Mostly we're at oncology meetings and infectious disease meetings. But natural products do have a role in CNS-related diseases, and I hope to tell you about that. I also want to express my appreciation to Drs. Lipinski, Behana, and Chico, because I won't have to talk about Rule of 5. I can just skip over my Rule of 5 slides, which will get us to the break quicker. So what are we talking about when we talk about natural products? These are not extracts of plants or materials that are mixtures. We're talking about distinct molecules that organisms, either microorganisms or plants or other eukaryotes make to cope with their environment. And here are some examples of natural products. This is aureomycin. This is actually the first of a series of tetracyclines. Many of you have probably taken this molecule or similar molecules for infectious diseases. This is fujimycin, FK506. This is a drug. This is was discovered to have strong immunosuppressive properties. It's a polyketide, as is this one here, which I'll talk about in a moment. What are polyketides? Both of these are made by Streptomyces strains, soil bacteria. This is palytoxin. This is not a drug. This is one of the largest natural products ever reported. It's made actually by this marine zoanthid. It has very, very profound pharmacology, hopefully that you will never encounter. It's a very potent channel blocker and very lethal if you're exposed to it. And then this actually is a very, very powerful drug. It happens to be not require a prescription, but the pharmacology of nicotine is something that many of us are familiar with. And I'll talk about how this actually has been a lead for a smoking cessation drug. That'll come at the end of the talk. So when we talk about natural products and their role in drug discovery, you can look at them a number of ways. And Dave Newman at the NCI has published a number of papers. And he, by his criteria, just about half of all the new chemical entities that were launched between 1940 and 2006 are natural product derived. Dave uses somewhat of a loose definition of a natural product. If you take a very rigorous definition, that would be molecules where the drug is actually the natural product molecule, the structure itself, or some derivative of it. You'll find that actually 58 out of, since 1970, 58 out of all the NCEs come directly from natural product, meaning they are the molecule itself or a semi-synthetic derivative of it. And that comes out to about 10% of all drugs launched in the last 10 years. Of these, as I mentioned, native molecules, there's 27 distinct natural products, the native structure, and 31 analogs. These sources, by far and away, microorganisms, and then plants, and then there's one compound from marine source. Here are the therapeutic areas where they impact, and actually this list is much longer, but basically all I could fit on the slide. But there are some unique challenges to natural products. One is accessibility. How do you manipulate these in the ways that Laura and Heather talked about in terms of optimizing their drug-like properties? They're challenging because they're complex structures often. If you're going to find new chemistry from extracts by screening, and I'll talk about that in a moment, that isolation is slow and resource intensive. And that's one of the reasons that Big Pharma has been stepping out of natural products actually over the last few decades. You can do pure natural products, you can screen pure natural products, but actually building libraries of pure natural products is very challenging in terms of resources. We heard a lot about chemical space. Actually, I can go quickly through this slide. Chemical space itself is virtually infinite. Actually, the number of molecules you can describe at molecular weight 500 and less is 10 to the 60th. So that's 1,000,000 to the 10th power, right? So virtually unlimited number of structures just in 500 molecular weight and below. But fortunately, not all chemical space is biologically relevant. So we're only looking for molecules that impact biologically relevant chemical space. And natural products, I hope to show you, are privileged molecules. They are biased to occupy biologically relevant chemical space because they're made by organisms, not by geological processes such as oil, petroleum components. So I'm also thankful we talked a little bit earlier about screening. So your standard HTS methodology is to find a target, right? This target you hope has some compelling evidence that it has some role in disease, and then you screen a large library of compounds, hundreds of thousands or even millions against this target, or fragments. You then find something that binds that target, and then you begin to optimize it by medicinal chemistry techniques. We're convinced that this is not the best way to screen natural products. We believe actually that they should be screened by phenotypic screening predominantly. And the reason is the following. If you have a biased library of molecules that you know bind biological targets or are predisposed to do so, you would want to expose those molecules to the widest array of targets possible, right? So in that sense, the widest array possible would be the living organism itself. And you use the phenotypic screen to do this. So you expose your library of natural products, your library of natural products, be it extract library or pure compound library, to a cell, and you get a phenotypic response. This happens to be a neurite outgrowth assay. The problem then, though, is what's the mechanism? And we really need to know how a compound works to advance it into the clinic. So in this black box of figuring out the mechanism, sometimes gives biologists the shivers because it can be a very difficult process. It can be straightforward depending on the disease, but it can be determining the absolute mechanism of action of a molecule can take a lot of work. But what you get out of that oftentimes are new targets. New targets perhaps with a natural product that found them is not necessarily a good drug, but that's a new target that you know has a phenotypic response. Right? So, and I'll show you some new targets that natural products has found in the past. So in terms of screening, if you're going to screen natural products, what do you screen? You have the options of screening an extract library, in which this case might be a library that's made by, let's say, a library of thousands of microorganisms. And any particular microorganism you'll see produces an array of compounds. I'll show you why they do that. And this is just an HPLC trace of a typical extract of a fermentation. You could inject some wine into the HPLC. You might see something similar that the yeast are making, in addition to a huge ethanol peak. But there's a vast array of molecules. The stuff up here is very polar stuff that tends to give HTS platforms the fits. And similarly, the stuff back here, greasy things, fatty acids, lipids of one sort or another. But the nice thing about screening this is that you expose your target to all these molecules, and your library is relatively small. You essentially have one extract per library. Here are the attributes and in terms of the library choice. You get the library size is small, assay interferences are high, sensitivity is modest, redundancy is very high because oftentimes the same molecules are made by many, many different organisms, different strains of an organism in your library. And your hit identification can be slow unless you have a good amount of resource to do that. You can pre-fractionate your library, in which you essentially cut it into fractions by some chromatographic technique. You toss out the problematic stuff because you can't make drugs out of those typically anyway. Sometimes you can. And then you have smaller mixtures. This relieves some of the bottlenecks here, but it also increases the size of your library. Typically, HTS, high throughput screening runs, you can do 10 cents a well. That's pretty good cost. So if you have a 1 million member library, the cost of doing an HTS is pretty significant. So this adds up quite quickly. But it does give the advantage of having a simpler mixture to work with. And then finally, you can screen pure compounds. This is the same fermentation. What I've highlighted in color here is which compounds you can expect to retrieve in sufficient quantity to put it in your library based on a 10-liter fermentation, which is the blue bars here, and a 100-liter fermentation. So there you have pure compounds, and there your hit is when you identify it, you have a distinct molecule. So I won't have to go into rule of 5 very much. The whole point of the rule of 5, as we've heard now in the previous 3 talks, is about oral bioavailability, because that's a very important aspect of a drug if it's going to impact the lives of patients. It's not absolutely essential depending on the therapy, but it's certainly very, very important for most drugs. It's an excellent strategy for many targets, but not targets involving protein-protein interaction. Some of the more complex biology of diseases we're working on now, Alzheimer's, Parkinson's, many neurological diseases that are poorly understood have to do with protein-protein interactions, or interactions of complex biology such as complex channels, DNA duplex interactions, whole pathway regulation. So if you look at the druggable genome, you'll see that we've learned over the years there are about 30,000 human genes. And about 3,000 of these are involved in some disease origin, disease-modifying effect. But this is the druggable genome. There are only about 3,000 of these 30,000 that you can actually go after with rule of 5 molecules. These are all best estimates based on what we know. These are not the same. So you really only have about 600 to 1,500 targets as estimated by Hopkins and co-workers in this publication of targets you can go after with rule of 5 molecules. But here, these undruggable biological targets are where many of the diseases are now that we need to treat. So we need molecules that will impact these other undruggable targets that are involved in disease. So why would we do this? Well, the track record is pretty good. These are all what we call beyond rule of 5 natural products, meaning these are molecules that don't go by Dr. Lipinski's rules, but they are still good drugs, and they are actually orally available. You'll see the oral bioavailability is actually pretty good, ranging from 100% down to about 20%. Some of these are the ones I described earlier. Here's cyclosporin, lipstatin, ivermectin. Miriocin is a new MS drug that came out from Novartis a year and a half ago. So these, rapamycin is one of ours. So these actually have, these are available. They have tricks about how they modify their conformations to get through membranes or to get through the BBB. So why screen anything for a hit in drugs, particularly natural products? At first, when you think about the numbers that roll up in terms of proteins, you wonder why we find anything at all, right? So if you take all the protein sequence space, it's essentially infinite. If you take a 300 amino acid protein, considering that there are 20 standard residues in proteins, you'll find that the number of possible protein sequences is 2 to the 300th. So that works out actually to 10 to the 80th, which is actually more than the known particles in the universe. You add up all the bosons, leptons, quarks. So the number of the protein sequences is virtually infinite. The whole complement of the estimated world proteome, all the proteins that are known, is about 10 to the 10. So what we know then is, by experience now, is that most proteins resemble other proteins. There are only about 100 protein domain families that are recognized by sequence. Since the papers that came out for this, maybe there's 50% more. And there's only about 1,000 distinct protein folds that you find so far that we've looked. All right, so there are many subdomain local rearrangements of secondary structures that are commonly used. And finally, there are biophysical constraints that limit the number of folded conformers. So actually, the protein fold space is much smaller than protein sequence space. Distinct sequences often adopt very similar folds. Highly similar sequences can adopt very different folds. Right? Unidentical peptide sequences can have very different conformation. Identical sequences can have very different conformations within different proteins. I'll show you some examples of this. So structure in the protein space is far more conserved than sequence space. So here's some examples. And here's how it relates to natural products. So these are 3 proteins. There's 3 proteins here. The first protein is an isomerase from Pseudomonas palustris. It's a microorganism. The second is a chain from an epoxide hydrolase from Rhodococcus, a different microorganism. And then the third is a polyketide cyclase module from a biosynthetic machinery of a microorganism. And what we've done is we've superimposed proteins 1 and 2. B is 2 and 3, and C is 1 and 3. And the orange areas are the stretches of structural homology. They're almost virtually superimposable structures. However, these 3 proteins only share about 20% of their sequence. So they're vastly different sequences, but you can see by the superimposition of the 3 proteins, they're virtually the same fold. So you would think that a binding molecule that binds one would bind the others, right? The converse is also true. Proteins with very high sequence similarity and have no structural similarity oftentimes. This one is actually BART from humans and an ADP ribosylation factor from actually fruit fly. They have 72% sequence homology. You can look it up in this publication. but they're virtually, there's no similarity whatsoever that we can see from the protein structure point of view. Similarly, you can make the same fold with various different ways. So you can see, look at these protein fold motifs. They're made in some cases by a single chain, two chains, or a single chain where two folds come on top of each other. But you can see that the fold motif is virtually the same. So the point here is that when organisms make molecules that they use for their own biology, it's likely that those molecules, because they bind protein folds, are going to bind other protein folds in different proteins and exhibit new biology. That's what we base natural products discovery on, that principle. So once you find a hit, how do you then take it to make it a drug? How do you modify the structure? So here are some examples. And actually, Heather, I believe, talked about some modifications of genistein to turn it into a drug-like molecule. And so I thank you for that, because I won't talk about semi-synthesis, modifying the core structure by synthesis. These are actually two molecules that are made by total synthesis. And this is myriocin. It actually comes from a fungus. And Novartis found it to be a very good S1P1 inhibitor, but it wasn't possible to take this forward. So they made fingolimod, which is Galenia, trade name. You can see it's very, very similar. And they've actually tied this part up into this benzene ring, and they get the same pharmacology and much, much better drug-like properties. This one, halachondrin B, this actually comes from a marine sponge, Halachondri okadai from Okinawa. And over the years, Eisai has now fully synthetically made this molecule, eribulin, which is a mitotic spindle poison, in a breast cancer drug. It was launched in 2010 or early 2011. Now, generally, when you show a medicinal chemist a structure like this, their eyes tend to roll back into their head because all these chiral centers, it's a daunting process to understand the SAR and the medchem, and then to make a molecule synthetically that actually is the right-hand portion of this. But Eisai, to their credit, the chemists at Eisai pulled this off, and while it was a long time to get there, they now have a completely differentiated antimitotic that works very, very well in the clinic as a single molecule. So another way you can do natural products, a way that we try to apply ourselves at Pfizer on this, is that these molecules are made by clusters of biosynthetic genes in the microorganism. And this is rapamycin. It's a drug. It's used to treat organ transplantation, immune suppressant. So polyketides, meaning many ketides. A ketide is a 2 or a 3 carbon carbonyl unit, are assembled in chains by these vastly huge collections of biosynthetic genes. And they're very modular. It's just like an assembly line. And they put these chains together, and then eventually the chain, each module adds 1, 2, or 3 carbon unit, and then the modules inside these submodules determine the oxidation state, whether it's a ketone, a hydroxy, a CH2, or a double bond. So you can see, actually, depending on how you put the modules in, you can get any kind of oxidative chemistry in each module that you put on, each building block you put on. That's how the organism gets a combinatorial chemistry effect. Eventually, these are then cyclized by another module. Well, it turns out that the starting unit, the first building block, is derived from shikimic acid. And actually, working with our collaborators at Biotica in the UK, we actually knocked out the biosynthesis of shikimic acid, and then you can feed in alternate starting units. And because the starter unit incorporation module is an enzyme, it will pull in, if it's the right structure, different starting units. And you can get modulation of the starter unit up here in terms of structural modulation. Equally, you can, if you're also clever and capable, you can replace modules. You can take modules out and put new modules in. You can delete modules. You can change the oxidation state by altering submodules of these. So it gives you because our tools are getting much better to manipulate genes in microorganisms, this is a viable approach now. So final few minutes, I want to talk about a couple of cases where natural products have become drugs. And the principles involved that I talked about earlier in terms of phenotypic screening and their privileged nature to bind protein folds. So this is a family of polyketides, many ketide units put together. Here's our old friend rapamycin. Here's FK506. They're actually the same family. You see, they share this structural motif here, as does this one. This is muridomycin and normuridomycin. But you see this part is different here, but they share this triketone unit here, and I'll show you why they do that. And then here's actually antascomycin. These are modest antibacterials. They have no immunosuppressive properties. These two do. This is a mild antibacterial. Antascomycin is a fascinating molecule, has the same triketone motif, but no one can figure out what it does yet. So rapamycin is one of these cases where you would find something by phenotypic screen that you could never find by designing a targeted screen, and here's why. So this is the complex. This is rapamycin here, and it was found early on that rapamycin was actually originally discovered as an antifungal, but it was discontinued because it was immune suppressive, right? Definitely an adverse effect that you would not be able to advance something clinically as an antifungal. But when it was investigated in the lab at Stuart Schreiber at Harvard in the early and late 90s,